Hello everyone, this is Ubaid Tariq, and in this session I am going to solve the problems or assignments of the course named Cell Culture Technologies, which is taught by Professor Manik Das on the NPTEL. So in this series I am going to solve the assignments that are given in this NPTEL course named Cell Culture Technologies, and in today's session I'm going to solve the week 0 problems or week 0 assignment of this course. Before solving the problems or assignments of this course, let me go through some of the basic principles of cell culture. So first, maybe in your mind, you may have the question, you may be knowing this, what is the cell culture? Cell culture is a technique to grow the cells in an artificial environment. So as you know, the cells in our body, they have some microenvironment, some environment outside the cells, outside. And this environment in our body, it is regulated. Its temperature is regulated, pH is regulated, oxygen and nutrient supply is regulated, and all this is regulated. So that condition uh, we have to produce that condition in an artificial environment in a petri plate. So we have to provide the ideal growth conditions for these cells in order to survive, in order to divide. So we have to provide ideal growth conditions. And in those conditions, what are these conditions? So we have to provide the necessary nutrients to the cells like amino acids, growth factors, glucose. These things we have to provide. Ideal temperature and pH should be maintained so that the cells will grow and divide, and there will be no stress on the cells. And this ideal temperature is our body temperature is 37 degrees centigrade, and pH is the physiological pH, which is 7.4. And optimal humidity is also maintained in the cell culture to grow these cells well. Now you may also have the question, why do we grow these cells? As you know that we have the organs or organ systems, and all these organs are made of muscles, and all this made of tissue. All these organs are made of some tissues. For example, in this case, is the muscle tissue, and this tissue is made of cells. So ultimately, the basic structure unit of the body or organ, it is this cell, and it is the functional unit. All this function, what this cell is doing, the same function this when these cells combine and the organ functions because of this particular type of the cell. So it is important to study the cell in an in vitro and artificial environment. To study, for example, to study the normal physiology. If you want to study some normal physiology of the cell or organ, so the basic unit is the cell, and we can easily study the cell in an in vitro condition to know the normal physiology of some organ or body. Study of the disease. For example, in the disease, some normal physiology is altered. There are some alterations. There are some changes in the organ or cell. So they can be studied from this cell. If there is a disease in some organ, so the cells of that organ can be studied to study what are the changes in the disease condition. These cells or the cell culture, it is also used for drug screening. For example, if someone wants to study some drug, some new drug, and its effect on the body or organ or some disease condition, so the cells can be this drug treatment can be given to the cell, and it can be easily studied. So as compared to giving a treatment to the whole organ or body, so but that is difficult to evaluate the effect. So in cells, we can directly check different markers after giving the drug treatment. Toxicity testing. For example, some drugs or some chemicals have the toxicity, and that toxicity can also be studied in these cells. Diagnosis of the disease is the same like the study of the disease. Like some cells have uh, the number of cells, or some cells have some particular markers, and the expression of these markers tell us about the disease condition. So they can be used for the diagnosis. these cells can be used for diagnosis of the disease in some particular disease. For example, in case of cancer, the cells they have some there are some there is some maladaptive change in the cells, but they can be used to the cell culture can be used to diagnose the disease also. And genetic engineering, as you know, that genetic engineering is a technology where some cell or genetic engineered products are made. Uh, so in the genetic engineering, the cells can also be used so that the product which is genetically engineered it can be made. For example, if a plasmid is introduced containing some the gene of interest which is inserted in some cell, and after inserting in the cell, this plasmid can the inserted gene can express, and some genetic engineering can we can get some product, or we can manipulate the cell, uh, whatever we want to do we can do using the genetic engineering. In genetic engineering we can also engineer the cell if we want to engineer it, and if we want to modify its function or something like that. So there are different types of cultures or different types of cells. So different types of cultures are also on the basis of different types of cells. For example, in case of adherent culture and in case of suspension culture, adherent culture are those cultures in which adherent cells or for example the cells, they have the property that they can adhere on the surface. They grow only adhered to the surface of the plate, because they have the property of creating extracellular matrix and that extracellular matrix because of that they adhere to the wall of the petri plate or the flask. And so it's also called anchorage dependent. They need to anchor on the surface, or they need to be clumped so that they can grow or they can divide, because they have the property in our body. For example, different epithelial cells or different epidermal cells. They actually they inside our body they also grow in an encourage dependent manner. They grow head, they go adjacent to each other, so they interact with each other. So in that case, here in the culture plate, they also go adherent in the colonies. In case of suspension culture, these are the cells which grow in the suspension. For example, different types of blood cells. So they do not adhere to the cell culture plate. They do not screen extracellular matrix. For example, the blood cells which flow in our veins or arteries. So they don't have the property to adhere for the growth. So here in the culture also, in vitro conditions also, they remain suspended. So another cell culture types are primary cells or cell lines. Primary cells are cells that are isolated from the organs, some organs or tissues, and they are directly grown itself. And cell lines, these are also the cells, and they may also be isolated from some organ, but they are modified so that they can grow, and they can be immortal, or they can go up to so many number of cycles. The primary cells actually they do not grow for too much passage. After some time they will be dead, because they don't because they although we are providing them the conditions, but because it is an in vitro condition, after some time there will be the damage to the cells, and after some time they will not grow properly. But cell lines are modified in such a way, uh, there is a genetic modification in this. So some genes are modified so that they will continue to grow even even at many passages. Now let's start the video assignment of the course. And there are some of the basic questions that are given in this. So first question is, cells that are dashed need to be cultured while attached on a solid or semi-solid surface. So already I have told you in the introduction, the cells that are anchorage dependent. They needs to be cultured on attached on the solid surface because they have the dependence. So they need to be attached to the some surface, and after attaching only they will grow only after attaching with the surface. So anchorage dependent cells or the cells that need to be attached on the single solid surface, and anchorage independent cells I have earlier given you the example that anchorage independent cells they, they grow in the suspension culture. They do not need to be attached on the surface for their growth. And this living and non-living is the option. Although non-living cells they do not attach even if they are anchorage dependent. If they do not attach to the surface, they get dislodged from the surface. But this living is a general thing, so this anchorage dependent is the answer. So next question is which of these chemicals is used as a protective agent when cells are stored frozen? So this storing cells is called, it is the term which is used for this is making the stocks. So we are stocking the cells for further use after maybe 1 or 2 years if we want to use the cells, or after some time we want to use the cells. So these cells are stored at a freezing temperature, for example, below 130 degrees centigrade. Usually, for some time, they are stored at minus 80 degrees centigrade, and after that, for a long time, they are stored at minus 196 degrees centigrade in liquid nitrogen. So, for storing the cells, we also add some of the components which are called cryopreservatives. So DMSO and glycerol or the cryopreservatives which are added to the cells which are to be stored, and they prevent the formation of ice crystals in the cells when if the cells are stored without these DMSO and glycerol. The ice crystals are formed in the cytoplasm of the cell, and it will damage the cell or will break the this cell membrane, and this DMS and glycerol goes inside this and prevents the formation of these ice crystals in the cell. So they are the protective agents or cryopreservatives. So the damage is prevented. After that, when we revive the cells will be revived or they will grow further. So next question is cryopreservation of cells is done at temperatures. So I was discussing the same with you guys that the cryopreservation or storing of the cells, it is done at below 130 degrees centigrade, and for the short term the cells are grown sorry, the cells are stored at minus 80 degrees centigrade for some short term for 1 or 2 days, but after that they are transferred in the liquid nitrogen, which is a temperature of minus 190 degrees centigrade. And here I will tell you one more thing that the when we first time this freeze the cells, that time we don't have to freeze them abruptly. For this, for example, if we took the cells in the vials, cryo vials, and place them directly uh, in the minus, at minus 80 degrees centigrade, this will also damage the cells because it will cause abrupt freezing. So for this, the isopropanol box is used. It's the box which contains isopropanol from outside, and inside it has some place where these cryovials are placed, and it helps in slow cooling of slow freezing of the cells so that they will not be damaged. Next question is aseptic techniques are required. Why we need the aseptic techniques? If you have worked in the cell culture, you may be knowing that the cell culture room is very well maintained, so that the aseptic techniques or sterility is maintained. So the laminar hood or basal two type hoods are used in the cell culture to prevent the air from outside to grow in the work to go into the working area. So so the main purpose of this is to prevent the contamination of to prevent the contamination in the cell culture, because if the cells are contaminated, there is also this to prevent only fungal attack, but not only the fungal attack. All the contaminations, for bacterial contamination or yeast contamination or some other contaminations, they all need to be prevented. And most common contamination is a bacterial contamination. So so the aseptic techniques need to be done. So aseptic techniques are required to prevent the contamination in the cell culture. You can also say that why we need to this aseptic techniques, and why we need to prevent the contamination. Let bacteria also grow, but bacteria will hinder the growth of the cells, or it will make the cells diseased, or cells will die. Like the bacterial infection in our body damage, the same damage the bacteria will do in cell culture plate. And in the cell culture plate, the media is very rich in nutrients and whatever it's, and so in that case they grow very faster and they will damage the cells. So in that case the contamination is very critical to be prevented. So next question is cells prefer to attach on. So what is the answer? So the answer is the hydrophilic surfaces. Cells usually prefer to attach on the hydrophilic surfaces because the environment outside the cell in our body is a hydrophilic environment, because the phospholipids outside the phosphate head, it is adhered to the water there, or but there's a hydrophilic environment outside the cells, so they also attach to the hydrophilic environment or negatively charged surfaces. Somehow they can also attach to the positively charged surfaces, but hydrophilic is the most important thing. So the cells attach to the hydrophilic surfaces only. So for this, the cell culture, these plates, they earlier they were made of the glasses. Now they are made of the polystyrene plastic. So they are coated earlier they were coated usually by collagen or fibronectin, other extracellular matrix components. So they help, they are hydrophilic and they help in the attachment of cells. But nowadays these polystyrene cell culture plates, they are treated by alpha poly lysine, so which gives the negative charge to this, and also the hydrophilic environment, so the cells are attached to this activity. So the answer is the hydrophilic surfaces. So next question is, which of the following molecules used for surface coating for cell attachment is of organic origin? So as I said you that the polystyrene, this glass, the glass or polystyrene, the plastic, these flasks or these plates, they are coated with some materials so that cells adhere to these materials. So this question asks what is the coating material, but that should be organic in origin. So here the organic origin, actually poly D lysine is also used in some cases like poly L lysine. So the organic origin here is the collagen. I already said that collagen is also used. Earlier it was mostly used uh, for for treating the flasks, so that the cells can adhere on the surface. So some flasks come as non-treated, so there is no treatment on the surface for the attachment, and in some come as some come as from the company, they come as treated, and the surface is treated with this quality lysine or collagen. And in if the surface is non treated, you can itself yourself treat in the lab with the collagen. Another question is cell culture media by the name of DMEM stands for. So the DMEM stands for Delbecque's modified Eagles media. So it is on in the name of Harry Eagle, which the name was given in the name of Harry Eagle, and he has discovered or developed this media in 1959. And the composition varies from maybe the general confusion or the general composition is usually 4 millimole glutamate and 4500 mg per liter glucose and some sodium pyrite, some salts like calcium chloride, potassium chloride, magnesium sulfate, sodium chloride, sodium phosphate, some folic acid, some different vitamins, and choline. These are the compositions which he have developed this media for cell culture. So he has also developed some acid, basal media, eagle, basal media, some alpha amino, essential media, different types of media he has developed. So it's on the base on his name. Delbecos modified Eagles media is mostly used in for different types of cell cultures. It's a basic media, but for some types of cells there are some specific media, or there are some specific requirements of the cells. In that case, some specific media are used in those types of cells. The question number 8 is phenol red indicator is primarily used in cell culture media for. So the phenol red, as you may be knowing, that is a pH indicator. So it's used to maintain the pH, or to not maintain the pH. Actually, the buffers are there which maintains the pH, which registers the change in pH. But so as the media is maintained at the pH of 7.4 or 7.3, so it is in the physiological range. So if there is a change in pH, this indicator of edit in the media, so that if there is a change in if there is a change in pH, so by visualization we will get to know that pH of the media is changed. For example, if the acids are secreted by cells and the media changes in the acidic, so it's not suitable for the further growth of the cell. So that we need to change the media. So if the phenol red in that case turns yellowish in color, so the pH of the media is reduced. So we need to change the media. Or in some cases, if some alkalis or bases are secreted, it turns into the pink color. And in the pH range of, I think, 6.8 to 6.8 to 8.2, it is red in color. So in the physiological range, it is red in color. When it goes extreme down, or it changes its color, so that we will get to know that there is a change in pH of the media, so that we can change it in the media. So the next question is, which of these cell culture media is carbon dioxide independent? As you may know, knowing that Dalbeco's modified Eagles media, DMM, is a carbon dioxide dependent media. What does I mean by this carbon dioxide dependent media? As you may be knowing that there are bicarbonates in the media, and they act as buffering agents, and there is the change in pH. And also the pH it is maintained because of the CO2 which is given to the cell. You may be knowing that 5% of CO2 is given, and it is as and this CO2 is not a metabolic requirement of the cell. They don't need this CO2, but it is used to control the pH. So the CO2 percentage is according to the bicarbonate concentration. They are related to each other. So when the CO2 is given, it gets dissolved into the media, and the CO2 reacts with water, and it forms the carbonic acid. And this carbonic acid, in presence of the amount of carbonic acid depends on the concentration of CO2 or percentage CO2 you are given. And the same it is the same concentration, or which is related to it, is maintained. The concentration of bicarbonate ions is maintained, which is related to this carbonic acid, and they both interact and form the conjugate base, and they act as a buffer system, and they resist the change in pH. So in case of maximum most of the medias, they are carbon dioxide independent, and only after giving the carbon dioxide the pH of 7.4 is maintained, and the buffer system acts only when the carbonic acid is made and the carbonic acid interacts with the sodium bicarbonate and forms the buffer. But there are some medias which do not have this bicarbonate thing, and they do not need. We don't need to give the CO2 to the cells. They are CO2 independent, and there are some buffers. And here the answer is L15, uh, L15 media, and this L15 media or L15 culture media, uh, it does not contain actually sodium bicarbonate. It has very little amount of sodium bicarbonate, and this for the maintenance of pH, we don't need to give the CO2. So but here also we have to maintain the pH, and here different types of different buffers are used. Like for example, sodium like phosphate buffers are used. Some free amino acids are used. Some galactose, some sodium pyruvate is used to maintain the pH. Usually, mainly the phosphate buffer is used to maintain the pH. But now you will say, why not in every media we use this for phosphate buffer, and then we don't need to give the CO2, and that a lot of resources will be saved. But the reason for this that is because in our body, in our physiological system, the bicarbonate buffering system is there in our body, several bicarbonates. So to mimic the physiology, to mimic the physiology, we have to give the bicarbonates, and with that we have to give the CO2. So we have to mimic this bicarbonate buffering system of our body in the cell culture. That's why most of the cases this bicarbonate or CO2 dependent culture media is used. But in some extreme cases where the bicarbonate is not desirable for the cells, or we have to, or some special cells, for example, I think some types of cells, I don't exactly remember the name, hepatocytes or HeLa cells or HEP2 cells. I remember now. They are the cells that need to be grown, or they need to be cultured with the CO2. So in that case, the L15 media is used, which is CO2 independent media. For the growth of the cells, or for the growth of the mammalian cells in the cell culture. Some of the non-defining components of the media are also added. For example, the fetal bovine serum. It is the serum which is derived from the bovine fetus, blood of the bovine fetus, and this blood is collected from the slaughterhouses, and it is collected in a closed system, or in the sterile system, and then it is processed, and it is used in the cell culture, and it is a non-defining media. It contains. So here you had answered what are the important things that this contain, and the answer is D. It contains the growth factors. It contains different growth factors which are essential for the growth of the cells, and they are not provided. Some amino acids and some growth factors, they are not provided in the Dalbecker's modified Eagles media, so we have to add 10% FBS to make the complete media in, so that cells will grow properly. And this was all about this week zero session of the cell culture technologies, where I give you some brief of the cell culture, and after that I discussed some of the questions which are very basic with you. So from the next lectures, I will be solving different assignments or different problems or questions with you, and apart from only giving you the answer, I will also try to clear the concepts which are related to that question. So that will help you to understand it more properly, and help you to clear the doubts also. Thank you for your attention. I will see you in the next session. Bye bye.